All right, thank you so much, guys, for coming. So today we're really cruising right along. We're going to do lecture 13, Synapse, and it's going to be a lot of information we've covered before because we talked about it when we did the neuromuscular junction. So today we'll mostly talk about the synapse between neurons, all right? And we'll talk about how, and more about neurotransmitters, and we'll also introduce neuromodulators, which control how neurotransmitters, send the message to the next neuron. And then we'll just talk about how they all connect together, all right, at the end, when we talk about neuronal pools and so forth, all right? So synapse, we have already talked about it when we talked with muscles that the neuron doesn't actually touch the muscle cell, right? There's actually a gap between the two, and that the connection is purely functional, in that the neuron can influence what's happening in the next cell, right? So, so that's what we'll do today. What happens is that the next cell could either be excited or inhibited, all right? And then we'll talk about those two scenarios, and then we'll talk about which ions are involved, which protein channels are involved when there's an excitation, and which proteins and ions are involved when there is a inhibition, all right? So synapse, of course, is the junction between two cells. It could be a neuron and a muscle cell, which we talked about quite a bit last time, or it could be between two neurons, all right? The cell that is the first one before the synapse is the presynaptic cell, just like the word means presynaptic, before the synapse, all right? And then the postsynaptic cell will be either the muscle fiber or the next or the second neuron, right? And we already talked about it. This is the functional connection, because these are not physically touching, and we talked about the advantages of having that functional connection where there's not a physical connection, okay? Because these are electrical signals. As long as the wires are bound together, whenever something happens, it's going to immediately impact the next cell, okay? And also, we won't have neuromodulators, okay? Because neuromodulators can modulate what happens at the synapse, okay? So that's a big advantage for that. So there's two types of synapse. One which is called an electrical synapse, all right? This one we haven't talked about, okay? An electrical synapse is when cells are connected by gap junctions, all right? And these gap junctions are protein channels that will let ions go through, okay? So if one of the cells is stimulated, then you're going to have the gap junctions passing on the impulse throughout the rest of the cells via these gap junctions. So it's going to act as if it's one membrane, okay? So that's what electrical synapse, and we'll talk a little bit about that and the kind of proteins that are involved too with the electrical synapse. So, those proteins are called connections, and we'll cover that, all right? So this is usually found in smooth muscle, okay? It's found in, what else is it found in? The heart as well, okay? It's found in the heart. That's my picture of that. It's found in the heart. Okay, where the heart is specialized connections called intercalated discs. Okay, and those are also same protein channels that will let ions through, so that when you excite one heart muscle, they all contract as if they are one cell. It's a functional system. So that's what electrical synapses has an advantage for. Okay, and then chemical synapse is what we've been talking about. The chemical is responsible for passing on the messages. And the chemical we've talked about so far in the neuromuscular junction is, it's the actual messenger that's sent to the next. What is the messenger that's sent to the next cell? ACH. Yeah, acetylcholine. So shortcut and beautiful. That's good. So ACH is the chemical, and we call it a neurotransmitter. Okay, so that's a chemical that's used to pass the message on, and here the message is passed on electrically via those gap junctions when the impulse goes through as if it's going through the same membrane. So chemical synapse, we already know what it is. So we'll talk about electrical synapse now. All right, and then we'll finish the rest of the time talking about the chemical synapse, because there's mostly a lot of chemical synapse, and then we'll talk about other neurotransmitters and some neuromodulators as well. Okay, besides ACH. All right. So like I was saying, the proteins that are, transmit the ion currents, which are really transmembrane protein channels, they're called connexons, all right, because they kind of sort of connect the cells, and they, which results in the passage of the impulse through all the cell membranes as if it was one. Okay, so they pass ionic currents, all right, such that the impulse will travel through the whole cells, all the cells as if it's one cell. All right. So you know how about the action potential, how it is propagated along an axon? Okay, I think in the modules, one of the problems you had was conduction and the sequence of events that follow an action potential. Some of them were a little hard, but we'll talk a little bit more about that. So what happens is the action potential happens in a certain part of the membrane, and then when it's happening, it will be spreading the current back and forth. Okay? Because the part of the membrane that's going through the action potential is in refractory period, the current will affect the next part of the membrane so that the impulse only goes in one direction. Okay, so those are the currents we're talking about here. Okay, so the currents will cause, the action potential happens here, and then the current will go back and forth like that, but this side is not going to respond to any more stimulus because it's in refractory period. So the impulse will only go this way, and then another action potential will be generated in the adjacent cell. Okay, so and then these gap junctions will help to pass the current through by allowing ions to go in and out, because the action potential is purely sodium in, potassium out, sodium in, like that. Okay, so that's what the connections will allow. So action potentials are then conducted very rapidly because these gap junctions or these connections will allow ions to pass in and out. So it will act as if it's all one membrane. All right. That's how muscles in the heart, muscle cells in the heart and smooth muscle cells will conduct as if they are one cell. Okay. Functional syncytium. So that's where the common cardiac muscle cells have that connections, and they are specialized in the heart, and they're called intercalated discs. And we'll talk a lot more intercalated discs. Those are the specialized cell-to-cell connections in the cardiac muscle cells. And smooth muscle cells have gap junctions. All right, gap junctions. Gap junctions, we talked about it. Remember when we talked about cell-to-cell connections? These are gap junctions. All right? So I want you to be able to tell me about electric impulses, electric synapses, and how they conduct through the cells as if it's one membrane, okay? And just be able to tell me about, some examples, like the cardiac muscle cells have those intercalated discs, and smooth muscle cells have gap junctions, and they have electrical impulses, electrical synapses. So now we move on to chemical synapses. Most of these synapses are chemical, and there's several neurotransmitters besides ACH. All right. So we already know about the synaptic terminal, right? The presynaptic terminal. It terminates in a knob like that. And then in that knob, what do you have? The presynaptic terminal, or the presynaptic membrane. You have these vesicles here carrying the neurotransmitter. For the most part, it's acetylcholine, but it could be norepinephrine, it could be epinephrine, serotonin, and we'll talk a little bit about that. And I'll give you situations where you have problems with those neurotransmitters, which gives to some of the psychological conditions that arise, and we'll talk about that and how they are formed, okay, towards the end. So presynaptic terminal contains the synaptic vesicles containing the neurotransmitter, right? Because it transmits the message, okay? And in the exam, we talked about the components of the synapse, right? And I asked you about, was it in this exam? I think it was, when we talked about the motor end plate and what it was, and you were supposed to describe that, all right? And then the presynaptic terminal, which has all these vesicles in there. And the motor end plate, what's special about the motor end plate? What's special about that? Remember, the motor end plate is the part of the membrane of the next cell that is close to the terminal, to the presynaptic terminal. Yeah, that's where the receptors. In the case we talked about, we said sodium ligand gated channels. Okay, all right. So whatever the neurotransmitter is, there will be receptors for it on that motor end plate. All right, and we said the physical gap between the two is the synaptic cleft, right?
Does every terminal have the multiple receptors for each of the different, or is some of them specified?
There's other neurotransmitters besides acetylcholine, and each of the synapses will have one neurotransmitter that it responds to. So certain cells are specialized to transmit only acetylcholine. Certain cells are transmitting epinephrine, norepinephrine, or serotonin. Okay, so it will only respond to that one, because on the motor end plate you only have receptors for that one neurotransmitter.
So in muscles, though, it's only ACH?
It's only ACH, yes. That way the body can regulate, say, ACH, then it knows it's going to affect the muscle cells. Okay? If it's no epinephrine, then it's going to affect, you know, how you speed up everything in the body. So it's going to affect certain glands and certain parts, you know. So only one neurotransmitter will work at that synapse. And there are many synapses and there are many neurotransmitters. Because one of the biggest things about the body is that it's separated and it specializes. Each part specializes in doing one thing. That's why it becomes good at it. You know what I'm saying? Imagine if you had like 10 jobs and you have to be this, you have to be that. You can't be very good at any one of those 10 jobs. Mostly we have just the one job and we become good at it because we're only just doing that one thing. So that's the same thing with the cells. Here they only, specialize in doing one thing. So we get that membranes, postsynaptic and presynaptic. So the postsynaptic membrane is one that carries the motor end plate. That's where you have the neurotransmitter, receptors. And then the synaptic cleft is the physical gap. So none of this is very new. It's not new at all. We already know about this. Presynaptic and postsynaptic are intuitive. They just pretty much tell you before the synapse and after the synapse. All right, and we've seen this picture before of the axons coming into muscles, and then going to each muscle cell, and you have the neuromuscular junction over there. All right, so we've seen that. So now, and we talked about events at the neurotransmitter at the neuromuscular junction, right? And we went through all that, and it was even in the exam, I think. But today we'll kind of talk about those events briefly and talk about what happens in the postsynaptic cell, okay? Because it could either be inhibited, which is inhibitory postsynaptic potential, and then it could be excited or stimulated, which would be an excitatory postsynaptic potential. So we'll kind of focus on what's happening after the synapse today. But briefly, we all know about how when the impulse comes to the synaptic terminal, it's going to affect voltage-gated calcium channels, right? And it is the calcium that goes in and then causes the vesicles to have exocytosis so that the neurotransmitter is sprinkled into the synaptic cleft. Okay, so we know about that already. So these processes, the basic processes, you already know that. And then the calcium ions, of course, diffuse in once those calcium gates open. All right, and then they diffuse in, and then when they diffuse in, they will attach to these vesicles, and it will cause these vesicles to attach to the presynaptic membrane so that exocytosis occurs, right? So this is nothing new, right? We've done this, and then the neurotransmitter, of course, diffuses through the cleft here, and it goes down there, and then attaches to the ligand-gated, channels. And the channel that is there will influence what happens. If it's a sodium channel, then you know what we did the action potentials. So we know what depolarization is, right? And you know which ion is involved, right? Okay, and then, so that's sodium. So if, the motor end plate has got sodium channels, so guess what's going to happen? What will happen? Sodium will go in, and then when sodium goes in, what happens? That's depolarization, right? So that means, are we likely to have an excitatory synaptic potential or an inhibitory synaptic potential? Excitatory. Okay. All right. If let's say it was a potassium channel, for instance, okay, and then the potassium channel opens up, what's going to happen? It's potassium channels this time. What's going to happen? Where is the potassium in the first place?
Inside.
It's inside. So where is it going to come out? What's going to happen? It's going to go out, and when it goes out, what happens? To the membrane potential. Yeah, it becomes more negative, which is called, it's repolarization, but it's going past resting, so that's a hyperpolarization. It becomes more negative. So when that happens, it means, is it going to be an inhibitory or excitatory synaptic potential? Inhibitory, okay? So inhibitory will take it further from threshold, because threshold is minus 55. If it's going more than minus 70, then it's going to be further and further away from threshold, so you're less likely to have an, so that's how they inhibit. All right, and another ion which we talked about a little bit is chloride ions. I want you to start to think about what's happening on the next cell now, so that when you come to it, you're already fresh in your mind. Okay, chloride, we said sodium chloride is the major salt in the blood, right? So whenever you find the sodium, you're going to find the chloride, okay? So chloride is what charge? Negative, all right? And chloride is going to be where? Outside or inside the cell? Outside with the sodium. So if we have sodium chloride channels here, what's going to happen? Chloride is going to do what? Get into the cell, and what's going to happen to the membrane potential? It's going to become more negative. It's going to hyperpolarize, which means it's going to be inhibitory. Okay, because I'm going to be asking you what are the scenarios and what are the ions involved, at the synapse, and what happening in terms of what's happening in the next cell. Because we're moving on now, right? We've done all this before. So depending on which channel is here, that's what's going to happen to that post-synaptic cell. Okay, so, and that's what we'll focus on today, okay? Number 4. So those are the things we've done before. So we are focusing today on this channel. And depending on what channel that is, and that will influence what happens in the postsynaptic cell. So we'll talk about that. And that was in your module too, in terms of determining the difference between excitatory postsynaptic potential and inhibitory postsynaptic potential. Okay, and those differences are the ones I just talked about. All right. So here's our little cartoon of the day for the first one. All right.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So actually, if I tell you what ion is on the postsynaptic cell, what ion channel, then you'll be able to tell me what happens to that cell. You see what I'm saying? You could do that. Just because you know what happens to where the ions are at rest, and then depending on the charge of that ion, you could either make it more negative, going down, becoming more hyperpolarizing, or you can make it more positive, going towards threshold. and therefore you're stimulating. You see what I'm saying? So that's really the whole thing about today's lecture. What's happening in the next in the postsynaptic cell. All right. So chemical synapse activity. Okay. So the neurotransmitter, of course, can be stopped in two ways. Okay. Remember, I told you already that they don't touch, and the advantage is that you can manipulate it. Okay. So one of the ways is you can reuptake. The presynaptic membrane can reuptake by endocytosis the neurotransmitter, and then you stop the whole thing. Okay? Or, like we showed last time, with an acetylcholinesterase on the motor end plate, so there will be an enzyme that can break down that neurotransmitter at that synapse. So you could break it down, you see? So you cannot modulate it like that if they were touching, because once it happens, because they're already touching, it goes. But here you can kill the messenger, or you can take it back. You can give them a cell phone call and say, hey, come back, man, I didn't mean it. You know what I'm saying? So that allows you to do that. Because sometimes I'll say, okay, what are the two ways that you can regulate the synapse? Okay, how can you stop the neurotransmitter? What are the two ways that you can stop the neurotransmitter at the synapse? Then you talk about that. You talk about reuptake. You talk about breaking down. All right? So those are the two ways you can stop the impulse. And I'm going to talk about several neurotransmitters. They fall into different classes. I'm going to ask you to know about 3 of them for the exam purposes. But then there's other things, neurotransmitters and so forth, and they've got different actions and so forth. So this is where we're going to start to talk about, so we have a synaptic knob here, right? And then we've got the next cell here. Okay, so what we could do, we could have a neuromodulator that can synapse, pretty much another neuron will synapse with a presynaptic neuron, okay, and can influence what happens, and then it may send more or less, of the neurotransmitter, so it can be modulated depending on the response you want. Or you can have another neuron synapsing with the postsynaptic cell, so that it could be slowed down, or it could be, the response could be increased. Okay, so that's the function of neuromodulators. Okay, which are substances, that neurons will be released to, to affect presynaptically or postsynaptically. Okay, so you see, this will be a presynaptic neuromodulator that's released by this neuron, and this will be a postsynaptic modulator after the event, and then you could either slow it down or increase it. Okay, so that's neuromodulators. You can see the difference between a neuromodulator and a neurotransmitter, right? The neurotransmitter is what is passed in the synapse, but the neuromodulator is what influences what happens with that neurotransmitter. Okay, it can either upregulate the message, or it can downregulate it or downplay it, right? Depending on what you want. That's another beauty of having this synapse, is that you can modulate it like that. You can even increase it if you need more, or you can decrease it if you kind of need less. So that's the function of neuromodulators. So I want you to know the difference between neuromodulators. I think this one will show you the transmission. Let's see.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
I must have liked this because I put it 2 times. Okay, so I let it play because it's short, Otherwise I would have killed it in a little bit. That would have neuromodulated it, But anyway, so I'll take it off next time, okay? So this is what I was talking about. There are several neurotransmitters, all right? So the first one we've talked about is acetylcholine, right? And acetylcholine affects muscles, right? Okay, there's a certain immunological condition where your antibodies will attach to the acetylcholine receptors, okay? And then they will be broken down as if it's a God, as if it's an antigen, a foreign antigen, okay? So what's that? Lupus is another immunological condition where you're attacking your own cells, but it's different from this one. This one affects the way neurons control muscles. It's myasthenia gravis. Myasthenia gravis. So what happens is that when you remove the motor end plate acetylcholine receptors, what happens is that you're not going to be able to do what? To stimulate the muscle. Okay? So the muscle is not going to be stimulated, and then you're going to be weak, all right? And you're going to be lethargic. Some muscles are important for respiration. You know what I'm saying? That would suck if you cannot increase and decrease the volume of your thorax so you can breathe in and out. So you could die because of that. Okay? So this condition is what I want you to know. This neurotransmitter, acetylcholine, and how it happens. Okay? The antibodies will attack those acetylcholine receptors, okay, which are the sodium ligand-gated channels, so that the muscles can no longer contract. And it could really be bad. Okay, so and you see, in a normal person, see how many of those, the membrane is even going like this to increase the surface area, so that you have more of these ligand-gated sodium channels, right, which are attached to acetylcholine. Look at a person with myasthenia gravis. You see, this is an antibody. This Y-shaped, antibodies are kind of Y-shaped like that. Immunoglobulins. You see how it attaches to the proteins and it starts to knock them out. And then the membrane only has a few of them. No matter how much neurotransmitter is in there, there's nothing, nowhere to bind. There's nothing, no gate to open. Okay? So that's what happens in addiction. Because addiction happens when you abuse cocaine, heroin, or whatever, some of those drugs, LSD. And what you're doing is you're blocking either the reuptake of this neurotransmitter, so that you are flooding this neurotransmitter with what? You're flooding the synaptic cleft with a bunch of neurotransmitter, right? So what does the body do? It says, oh my God, we've got too much of these messengers. So what we do is we kill the receptors. So it starts to reduce these receptors, and then you need more, right, to have an effect. So that's why you end up overdosing. See what I'm saying? Because your body is saying, oh my God, we just overstimulated. Okay? And we'll talk about some of those neurotransmitters, okay? So, gotta know this condition that happens when you kill those acetylcholine, ligand-gated channels, myasthenia gravis. And it's caused by those antibodies. Your own antibodies somehow mistake them for a foreign antigen and they start to knock them out.
Is it genetic, or is it just something you can get upon?
Yeah, it could be genetic. You could have a protein that goes wrong. It's a autoimmune disease. Autoimmune disease occurs. We'll do this later on in the NP2, where when your body is producing these antibodies, it can produce antibodies at a rate of 2,000 in 1 second. Okay, if that doesn't amaze you, nothing ever will, because that's a long protein, and you're making 2,000 of them in 1 second. So you're going to make mistakes, right? So the body has checks and balances. It will either, knock off all those that will attach to cells of the host, your own cells. Sometimes that process goes a little bit, and then they survive. You let those antibodies that attach to your own cells to survive. So those antibodies will start to attack your own cells. So that's what will happen here. So it's an autoimmune condition, okay? But I want you to realize that it will cause muscles not to contract, okay? So that's what happens. Okay, so we have another neurotransmitter. This time we haven't seen that. Serotonin is a biological amine because it contains this amine group right here. CNH2. Okay, and this is normally inhibitory. Okay, so when you're depressed, most likely you overthink things. You know what I'm saying? Your brain is overstimulated. You kind of see something and you kind of blow it up out of proportion, and you become depressed, right? So there's serotonin, which is a neurotransmitter that inhibits that activity, so that you're not overwhelming yourself with too many things, right? So serotonin will inhibit that activity, and what happens in this condition is that when you're depressed, you don't have enough serotonin to inhibit that activity that causes you to become depressed. Okay, and so you have the antidepressant therapy, which are called selective serotonin reuptake inhibitors, SSRI. Okay, so they will prevent. Remember, at the neurotransmitter, we said the choline is sympathetic back into the for recycling by a sodium symport. Same thing happens with serotonin. Okay, it is also sympathetic back into the presynaptic cell via the sodium symport. Most of these neurotransmitters are sympathetic by the sodium symport. Okay, so what does the selective serotonin reuptake inhibitor do? It will inhibit this sodium symport, so that you have more serotonin to do that inhibition. Okay, so you see, you can have a neurotransmitter that has inhibitory function and still causing a positive result. Okay, and you can have a neurotransmitter like acetylcholine that excites and also causes a positive result. You see what I'm saying? So serotonin happens to inhibit some of that mental activity that will cause you to be depressed. So when you're depressed, you need to have more serotonin in there to do that inhibition of that activity that is causing depression. So what do? You block the sodium symport that symports it back. So you prevent the reuptake. Remember, one of the ways to stop a neurotransmitter is to reuptake, right? To take it back into the presynaptic cell. So I want you to know about serotonin as well. So that's another new, and how it is involved in depression. Okay? And it's mostly inhibitory. Okay? Serotonin is also a biological amine because of that amine group. Acetylcholine is
When
just...
someone's depressed, is it that it reuptakes it too fast before it does its work?
Yeah.
Or it doesn't release enough?
It could be one of those things. It could be any one thing just causing serotonin to be low in the cleft. Maybe it reuptakes it fast, or maybe it's not producing enough, and then some of it goes back. Okay, so in order to increase the amount of it that is in the synaptic cleft, so that it can attach to the receptors, you block these reuptaking, symport. So it's selective serotonin reuptake inhibitors, and then so that you have more of it remaining in there. Yeah, because the etiology could be one of other things. Maybe you're not making enough serotonin or something like that. You have less serotonin, so the less that you have, you just make sure that it's not taken back up by the cells. But one of the bad things about this is that it's very, you get used to it quickly, you adapt to it quickly too. Like Prozac is one of those drugs, and then soon enough you need another drug, because then again your body will start to regulate the receptors. You see what I'm saying? That's what it will do. Okay? So selective serotonin reuptake inhibitors will cause more of it to be in there, the serotonin. Okay? So gotta know myasthenia gravis, I gotta know the antidepressant and how therapy serotonin is involved in it. Okay? So you see what happens with serotonin, right? Right, very good. Okay, so serotonin is continued. All those other anxiety disorders and so forth, the selective serotonin reuptake inhibitors, they do the same thing. And drug addiction is shown by, by the fact that cocaine, blocks those dopamine, because dopamine is another neurotransmitter. But dopamine has two functions. Dopamine is either excitatory or inhibitory. Then when you have a neurotransmitter that has both functions, it's hard to treat conditions that are involved with it. Because dopamine is involved here, okay, and it's the excitatory part of it. Dopamine is also involved in Parkinson's disease. And in Parkinson's disease, it's the inhibitory function of dopamine that is needed. Because what's happening in Parkinson's disease? You're shaking too much. Because right now, as you're sitting right there, you are, because this is all electrical things, right? So your muscle can be stimulated because it's an electrical thing. So because there's lots of messages that are being passed up and forth. So there's normally this excitation that is not necessary because you want your arm to be still. So the inhibitory part of dopamine will inhibit that unnecessary motor activity. So when you don't have enough of that inhibitory dopamine, what happens is that that unnecessary motor activity is going to go on. Especially when you're trying to do something, that's why they have an intention frame. Okay? Because when they're really trying to do something and you're trying to send impulses, then you have all that noise also affecting the movement. Okay? So it's again because of the blockage of those reuptake, of those molecules. Okay? You see what I'm saying? So if you're taking cocaine and you're preventing the reuptake of dopamine, and then the dopamine is staying in the synaptic cleft more, your body is going to start to knock off the receptors again, just like it does with the selective serotonin reuptake inhibitors like Prozac and them. Because there's just too much serotonin that stays on board, your body will reduce the receptors, and then that's how you become addicted, and you need more and more dose, or you may have to hit it somewhere else in order to achieve the same effect.
So because people who are on cocaine, they're releasing more dopamine, that's why
they're that's why they get that excitatory thing, and they're all because you know what I'm saying, they become high. The first times it's okay, it's all good, right? Because your body hasn't knocked off the receptors and you become high. And then if you continue to abuse it, but this depends on every individual. Addiction depends. Some people don't get addicted easily, some do. Because you are your own. There's no other person like you, Spring or Tina. You see, you're the only person like that. Even twins, if they grow up in different environments, they can become different things, even though they have the same genetic material. Okay.
Go ahead. Okay, so like if there is an abuser, don't they override? Isn't there an overflow that receptors can't be messing up?
That's what happens. When you get too much dopamine in there, what's going to happen is that your body is going to start to knock off the receptors. It's going to say, my God, I'm overstimulated. So it's going to knock off the receptors. So it doesn't matter how much dopamine you have on board, there are no receptors. There are no gates to open. So what do? You inject more. That's why they end up overdosing. Because they just wanting to inject more and more and more. And it's individual basis, it depends. Some people get addicted, some don't.
And that's
just
due to the
depends.
dopamine that they already are producing?
Well-
or one person won't get addicted and one will?
Well, it just depends on your body chemistry, So that's why those drugs are hard to kind of modulate. Even to get drugs that go to the brain. Remember I talked about last time the blood-brain barrier stops almost anything from going up there. So the way they treat some of these things like Parkinson's disease, what do? They have sort of molecules that mimic dopamine. Sometimes they use L-dopa, which then causes production of more dopamine. Okay, because only dopamine will be accepted up in the brain. It's not going to pass. If you have another molecule, even though it does the function, it's not going to go into the brain to affect the activity of those neural receptors. Okay, so that's the dopamine and the serotonin, and the serotonin is inhibitory, but this one, dopamine, is both. The cocaine part of dopamine is the excitatory part that gives you a high. Okay, the Parkinson's disease is the inhibitory part that inhibits unnecessary motor activity. Okay, and the same deal happens with the drugs, is that you try to prevent the reuptake of that neurotransmitter so that you have more of it. Okay, so that's what cocaine will do, to block those reuptake of the dopamine and cause you to, and it's a symptoter as well. Those things usually piggyback on symptoters like for sodium and things like that. They all do. Acetylcholine does it, serotonin does it, dopamine doesn't. Okay, so I want you to know about the acetylcholine and the myasthenia gravis. I want you to know about the serotonin and depression. I want you to know about dopamine and cocaine, which is the excitatory part, dopamine and Parkinson's disease, which is the inhibitory part, which it inhibits unnecessary motor activity, okay? And then it's all about preventing the reuptake of that neurotransmitter back into the presynaptic cell, okay? So, some of the other ones like norepinephrine, this is the fight-flight things that will cause overexcitation. Like in ADHD, you've got overexcitation of that you can't focus. You're just too busy up in there. You cannot focus anything. You have amphetamines and so forth that will kind of try to increase the norepinephrine to knock off that activity, right? Okay.
It's like a reverse effect, right? Someone who's not ADHD takes amphetamine, they're going to be like... Oh,
they're going to be like, they get euphoric.
Right.
That's the high they want.
But someone who's already all wired...
They get toned down to normal. You see what I'm saying? So people abuse like painkillers, right? Painkillers. Because painkillers, they prevent the substance P, which is involved in pain. So if you are removing that pain, it gives you relief, it gives you a high. Because it knocks off, it makes you think there's no pain whatsoever in the whole world and you're just like up in the high, like that. You know what I'm saying? But that comes with the price. After a while, you start to remove those receptors because there's just too much on board. That's how the body deals with too much stimulation. It will just remove the receptors. And then it's hard to replace them. So that's why when you go into rehab, it takes a long time to shake it off. Because then you have to, get back those receptors and so forth. It takes time. Things like barbiturates, they kind of inhibit, and one of them is GABA. Okay, this is the last one I'm going to really talk about. Gamma aminobutyric acid. Okay, it causes, it can be used for anesthetic because it will reduce your activity. It will kind of, you see, this is an inhibitory molecule that is affected. It's got chloride channels. Remember chloride channels, we said if there are chloride channels, you inhibit. So it kind of puts you, it knocks you down. It's used as an anesthetic. So, a barbiturate will attach to that chloride channel so that it stays open more and more chloride goes into inhibit. Because when you get a negative charge into the cell, you hyperpolarize. Okay? So gamma aminobutyric acid is normally inhibitory. Okay? And when you drink beer, alcohol, it does the same thing. It gives that kind of, the high you get is because it's kind of making you, it kind of puts you low. So that's why sometimes you end up passing out if you drink too much. You know what I'm saying? Because you're kind of knocking yourself out.
That's why it's like if you're stressed and you drink a beer,
you get relaxed because it kind of it makes you think that nothing is going on, it's all good, and you don't think about your problems. So that's how it works. Okay, so that's the barbiturates. I knew you'd get a kick out of the alcohol part. But so it inhibits like that. So barbiturates do that, and some of them are used to anesthetize you. If you get enough, it knocks you completely down. Okay.
So that's what they use in like when you have surgery.
Yeah, when you have surgery, all those anesthetics, they pretty much just inhibit all that activity in you, and then you just knock off and you sleep. Okay, and then they do their surgery, and then they wake you up. All right. So that's the same thing with alcohol. Okay. You could also know about the GABA, but really the main ones I want you to is the serotonin with the depression, and then the acetylcholine with the myasthenia gravis. Okay. And then the dopamine. All right. That's involved in Parkinson's. That's inhibitory, and then the dopamine that's involved with the cocaine in drugs, that gives you a high, is the excitatory function of dopamine. Okay, so it's hard to regulate dopamine because you don't know whether you're going to increase the inhibitory effect or the excitatory effect. So you can't screen between those two. It's just the body. So that's why it's hard to treat some of those conditions. All right. There's strychnine poisoning and so forth. It causes your muscles to stay contracted and so forth. Some of these you can read for interest's sake, just to see what happens with this. You know, so that you know, you can just get an appreciation of some of these. And glutamate is another one that's involved in the brain, and it's excitatory. It's involved in stroke and things like that. When your brain doesn't have enough oxygen, and it tries to revive the brain by, you know. So anyway, so I want you to know about the first three really. And GABA. GABA is an inhibitory amino acid, gamma-aminobutyric acid. And then the other ones you can just read for, you know, just for your interest, because there's a lot of them, okay? There's a lot of neurotransmitters and lots of neuromodulators. This is the pain substance P, okay? And endorphins are natural painkillers. So your body has some endorphins that reduce pain, but if you take extra exogenous painkillers, then you get that high because it sort of puts you in a, so that's what all those opiates do and so, all right? And the nitric oxide is it's a gas that you know, causes vasodilation and so forth. Okay. Anyway, so I'll stop there, but know about the first three, okay? Those are the ones I might ask about in the exam, just to talk about myasthenia gravis, you know, Parkinson's disease and depression. And then, of course, the cocaine, you know, addiction. All right, so now we come to the two events that can happen. We've already sort of talked about it because we know the ions and the effect they cause, whether it be depolarization with the sodium or hyperpolarization with the chloride and the potassium ions. Okay. So an excitatory postsynaptic potential can only happen one way, right? And that's by affecting what channels? Sodium channels. Only sodium causes depolarization, you know what I'm saying? So that's why only sodium is going to be involved here for excitatory. So anything that's going to excite is going to affect sodium channels. Okay? That's going to be an excitatory postsynaptic potential. All right? And these are graded. Graded simply means that the response is proportional to the stimulus. Okay? So if it's you know how the membrane will be at rest like that. If it's a small one, it will just go like that. And then the thing about it is that it will start to disappear along the membrane. And then it goes back to normal. Okay? And then this one is maybe bigger, but if it doesn't reach threshold, still nothing happens and it disappears. Okay? So that's one thing about excitatory postsynaptic potential and inhibitory postsynaptic potential, is that they are conducted in a decremental fashion along the membrane. Okay? Because if it goes to threshold, you have an action potential. Okay? Because the module was asking you about that as well. You know, what happens with these excitatory? What are the similarities and differences? Okay? So one of the similarities is that they are both graded, and they both conducted in a decremental fashion along the membrane, and they just disappear. All right? The difference is that one causes depolarization, the other causes hyperpolarization. Okay? So, which is the inhibitory one. And the inhibitory one can happen two ways, right? We talked about how if you've got potassium channels, because there's more potassium inside the cell at rest, it's going to come out. It always goes down to concentration gradient. And if you're taking a positive charge out of the cell, it's becoming more and more negative. So that hyperpolarizing, which means you are moving further and further from threshold, which means you are less likely to have an action potential. So you're inhibiting. Okay? Because in the exam, I will ask about the two scenarios and the ions that are involved for each, in terms of the ion channels, right? For each of those two, excitatory and inhibitory. So presynaptic inhibition decreases neurotransmitter release, all right? That's in a neuromodulator kind of fashion. You know, you can reduce the amount of neurotransmitter that goes there so that you only get a little graded potential that probably doesn't do anything because it won't reach threshold. So you inhibit. presynaptic facilitation, you know, this will be in terms of neuromodulator, okay? Can upregulate it. You may have something that's going to cause just a little graded potential, but if you have something that will increase the response, you might reach threshold, and you have an action potential. So this is all the neuromodulation part, okay? Because neuromodulation, and at the end I'll show you a neuron. Remember, you can have thousands and thousands of synapses. Like, for instance, a presynaptic cell can synapse with about 1,000 other neurons, right? Okay, and then the average postsynaptic neuron can synapse with up to 10,000 other neurons. Okay, so that means it has 10,000 branches. And there are some parts of the cerebellum in the body that it will synapse with like 100,000 neurons. If you can imagine that, just try to wrap your brain around that. Okay, so that means that one neuron will have 100,000 branches. That's amazing. I can't even think about 100,000 now. To count, you know, 1, 2, 3, you run out of fingers real quick. Okay, so that's the neuromodulation part. You can affect it by increasing the response or decreasing the response. And I'll show you a nice picture where you have those neuromodulators. Okay, so here, if you have a depolarization, you see how you are approaching threshold. So that's an excitatory postsynaptic potential. It may not necessarily lead to an action potential, because if it doesn't reach threshold, then nothing happens. If it doesn't go to -55, nothing happens. All right, and then the membrane is at rest, and you apply something that's going to cause maybe potassium channels to open or chloride channels to open. Then you have hyperpolarization. You start moving further from threshold, making it even less likely to reach it, because you are now further down there. So you are inhibitory postsynaptic potential. Okay. This is the neuromodulation I was talking about. So you can do it presynaptic. Okay, you can see this neuron. You okay, Maria? And then, did you have a question? Yes, something exciting. So, yeah. Okay, so what happened here? This is also another terminal that can release. You see how when it's not releasing anything here, you've got a whole bunch of neurotransmitter that's being released here, and then over here, you start to modulate how this is going to respond by influencing how many calcium channels open here. Because it's the calcium that causes these guys to have exocytosis, right? So you just influence, if a few of them open, then a little bit of calcium gets in here, and then you only release a little bit of neurotransmitter. So you can modulate it, you can upregulate it, or you can decrease it. You can decrease it. So this is neuromodulation, okay? At the presynaptic. You can also neuromodulate postsynaptically too, okay? Anything, so these are those two things. Remember, excitatory postsynaptic potential can only be sodium, and this inhibitor can be the acetyl, can be the chlorine, chloride ion, and the potassium ions, okay? So, you see, that's sodium, and then that's didn't reach threshold, so nothing happens, okay? So another thing you'll see is that they can be summed up, okay? Because remember, if one of them is getting like 1,000 of these synapsing on it, what happens is that you could have an excitatory postsynaptic potential that doesn't reach threshold, and another one that doesn't reach threshold. They can add up, and then it can reach threshold. And I'll show you that in a little bit. So this is another one right here, where you have an inhibitory one, chloride gets in, and then it becomes more negative because it's negatively charged, right? So that's inhibitory postsynaptic potential. And then this one here is the same. It's the same inhibition, but this time you have potassium. And potassium, because it's more inside, it goes out, and then it still has the same effect of hyperpolarizing. All right? So, and that's it. Okay? So really know about those three ions and how they cause inhibitory postsynaptic potential and how they cause excitatory postsynaptic potential. All right? So remember, I was saying, because there's many of these neurons synapsing with many other neurons, what happens? You have, and they can summate. So the first one is spatial. Space is, you can think of it as having a neuron in this space and a neuron in that space, and they both firing at the same time, right? And then you have a little blimp and a little blimp from that one. Those little blimps will add up, and then you can reach threshold. Then you have an action potential. So that's spatial summation. Okay? And then temporal. Temporal means time, right? So this is one firing, and it's firing two action potentials close together. So you have one come in, and while it's still depolarizing or doing whatever it's supposed to do, another one comes in, and then it adds up again. So that's temporal summation. So temporal summation, one neuron is firing, and the frequency is high. So one comes in, and another one follows immediately after it, and before this one has had a chance to go down and rest, another action potential comes from the same neuron, and then it goes to spatial. Okay, so you see the difference between spatial and temporal. Spatial, we have two of them, or three neurons that fire a little potential that's graded, and another one, and those add up. That's spatial. You have several neurons. Temporal, you have only one, but it's just firing at high frequency. So that, yeah, one action potential after another, they're so close together that the postsynaptic cell doesn't get a chance to go down. And then it gets hit again while it's going, up, and then it continues to go up and it can reach threshold. So another way I will ask you is how these excitatory postsynaptic potentials or inhibitory postsynaptic potentials are, summed up. So there's spatial summation, there's temporal summation. Okay? This is the same neuron, and this is a few, and that adds up, and you reach threshold. So know about that. So inhibitory and excitatory neurons, they can converge. So that means several come to a few. Converging is coming together on one thing. Like maybe three neurons come together on one. Or it could diverge, right? Because it's got many terminals. One could diverge onto many. You see what I'm saying? One can control many. Or many can control one converging, or a few. You see what I'm saying? That's how these action potentials are arranged, so that you have several neurons doing something, and I'll show you the actual situation that happens in your body. It's not just one neuron firing and then something happens. It's several of them firing and modulating the events. So you may have more excitatory postsynaptic potentials than you have inhibitory postsynaptic potentials, and then you have an action potential. Or you may have, in some situations, more inhibitory postsynaptic potentials, less excitatory, then you have an inhibition. Okay, so I'm going to show you the two ways in which these action potentials influence one or several neurons. Okay, and that's via convergence and divergence. Okay, so this is what actually happens in the real world. Okay, this is just an example of spatial summation. Spatial summation, you have several neurons firing, and then they are causing an action potential because you end up reaching threshold, because they can be added up, those little graded potentials. And then temporal summation, that's when you have just the one, you see it fires an action potential, and another one immediately follows behind it, so that, you see, before it even came down to resting, another one hit, and then you get an action potential because it will reach threshold. Temporal summation is one neuron firing and the high frequency, all right? Action potentials are closely following each other so that they can summate. Spatial is several. They fire a little action potential and another one fires another action potential, and those two add up, and then you may reach threshold and have an action potential. So what's happening in real life, right? It's not happening all in isolation, okay? You have several. So you have a whole bunch of these neurons synapsing. Some of them may be firing excitatory, some of them inhibitory. And the sum of those two parts, like I was saying, if you have more excitatory than inhibitory, then you have an excitatory synaptic potential happening in the postsynaptic neuron. If you have more inhibitory than you have excitatory, that's what actually happens in the real world, because one will have several of them synapsing, and then it will have, some will be having spatial summation, some of them will be having temporal summation, some of them will be firing fast, some of them slow, things like that. So that's what happens in the real world. So this is what happens in the real world. Okay? It doesn't happen isolated, one at a time. No. Okay? So that's just what it shows. Okay, so again, so it leads us to neuronal circuits, right? I talked at first about the convergence, where you have several neurons coming and converging with a few, maybe one or two. Okay, so you have many coming into and then synapsing with one. So that's a convergent pathway, and I'll show you nice pictures. Okay? In the exam, I'll ask you about these neuronal pools and how these neuron circuits are arranged. Okay? So divergence, you have one or a few neurons synapsing with many neurons. So it's diverging, it's coming out like that. Okay? So that's a few will be affecting many. Okay? So that's a divergent pathway. And I'll ask you to describe these pathways and these neuronal circuits in the exam. I usually do. Okay? I'll always ask you about how, you know, the excitatory postsynaptic potentials occur, or the inhibitory postsynaptic potentials occur. And I ask you about neuromodulators and how you get summation of these potentials. And then oscillating circuits. Remember when we talked about the neurons, what did we say about the neurons and the axon? We talked about those branches. Remember those branches? Those branches at right angles to the axon? What did we call them? Does anybody remember what we called those branches? Well, the dendrites are on the cell body. I'm talking about the axon. There's sometimes a branch that is at right angles to the axon. What did we call it? It starts with a C. And it was a Tom Cruise movie that happened in Liquid. It starts with a C. Collateral? Collateral. Yeah, you know what I'm saying? I just remember that movie because it was the same name as collateral. They are called collateral branches. So this happens in the postsynaptic neuron, okay, and it will have, so this is the cell body, right, with the dendrites, and then it will have, okay, and then the terminals. So this collateral branch will come back and synapse with the presynaptic neuron. So that forms like a circle, right? So that's a, you know, that forms the oscillating. Oscillation is circular. It's going in circles. You see what I'm saying? So that means what has happened postsynaptically here can come back in a negative feedback fashion, kind of sort of inhibit. Like, let's say you've had enough of that stimulation over here, right? So these collateral branches will go back here and start to inhibit this neuron that's sending the information, okay? So that's an oscillating. So be able to explain and describe these neuronal pools, okay? Okay, so those are there. You see what I'm saying? You got the inputs here, you know, converging onto one. And then here, you've got, you know, divergent where you have one, and then it's going to many. And then here, oscillating, you see this collateral branch comes back and synapses with the presynaptic and also synapses with the neuron itself, okay? So those are the synaptic branches. Thank you guys, I appreciate it. Do you guys have time to do those midterm feedback forms? They help you to do your thing. I'll eat a little bit of your time, your break. So I apologize in advance. Otherwise, we won't get time. So I'll give one of you guys to give a few. And give me the remainder, okay? Because I have other classes. Can you do that for me, guys? You guys are like, we want our 15 minutes. Huh? Because then we won't have time, you know, I use this information, it really helps me to help you better and so forth. Let me know what's working and so forth and some of your favorite things. Okay, guys? Thanks. I appreciate it. I might as well give you more because if you have less, and then give me the remainder, please, so I can give to other classes. Thank you, guys. I'm supposed to exit quickly so that I don't sort of, you know, influence you guys. Oh, anyway.